Good morning, everyone, and well, I welcome you once again to our Kona Lab Talk, which are these webinars that we prepare from throughout the year for you. And this time is a turn of an ISO standard that I see has generated quite an interest. So, and although it is not really a method for analysis or anything similar, but rather for the issues of validation and verification of a method. So again, I thank you for your time, your interest. My name is Aletia Cruz, and I am a product manager for microbiology at Conalab. So we're going to start with the presentation, verify alternative methods in your lab according to ISO 16140, part 3. And as you can imagine, there is a lot of information to cover since this ISO is only one of 6 parts that make this series up. So, so some people are still joining, but I'm going to begin with the presentation because it's quite a bit of information. So let's start. So the key points that we're going to go over today is going to be, of course, everything and anything about ISO, and we will have to start talking about the series of ISO standards that I've mentioned, of which this part 3 is a part of, and what we can find in them. Then we will go into what validation versus verification is in itself, to really understand what each of them brings us and thus helps us to better understand verification itself. We will see the characteristics that we must determine in our laboratory for the method to be verified, and well, finally, conclusions that we take after all of this learning. So, as I mentioned, we're going to start with the series of ISO standards to have a better idea about the information that we can find in these documents and which part we have to go to depending on our objective. So there are 6 parts that make up this series of ISO 16140 standards. We start with part 1, in which we will mainly find all the vocabulary, terms, definitions that will help us better understand the rest of the parts of the standard. In part 2, we can find the protocol to follow to validate a proprietary alternative method that has been compared against a reference method. Both of these 2 first documents were the ones published in 2016. Then we have part 3, which is what we will focus on today, the protocol for verification of a reference method, of an alternative, of a reference method or an alternative method validated in a single laboratory. It is the most recent standard published just 2 years ago. The following parts are protocols for validating internal methods, for interlaboratory factorial validation of non-proprietary methods, and for validation of alternative methods for confirmation and typing. So we have the ISO 16140 series of standards, and in it we find protocols for both the validation and the verification of different methods. For the verification, if this is what we are looking for, then we would go directly to part 3, which is right here, and that would be the only one that would apply to perform a verification. Then for the validation, depending on whether what we're looking for is a qualitative or a quantitative method, we would have to be clear if it's a reference method or an alternative method. So if it's a reference method, we would look at ISO 17468. And on the contrary, if it's an alternative method. Again, we have to check, is it a proprietary method, a non-proprietary method? If it's the first, then we would consult part 2. If it's a non-proprietary method, again, we have a question. Is it for a single laboratory or for multiple laboratories? So if it's for a single laboratory, then in our case would be, we would go to part 4, or for multiple laboratories, which would be part 5. Finally, and if in what we are interested is I forgot about this part, the validation of a confirmation or typing method. So it's only for alternative methods, and that would be part 6. So having said this, parts 2 and 6 would actually be used by certification bodies to carry out the validation of these methods. And on the other side, part 3, 4, and 5 would be for the verification or validation of methods in our laboratory, which the responsible would be the user laboratory. It seems to me that with this diagram, it is very clear which protocol we have, we can find in each of these parts of this series of standards, and which one we should address according to what we want to do in our laboratory for a particular method. To understand more about what is validation, verification, a reference method, an alternative method, we have added a document. You have in your GoToWebinar box, you have documents, and there you can download a document with the vocabulary we have, well, with key concepts we have prepared for you, so you are able to understand it even better. So continuing. Why is this series of ISO standards so important for a laboratory that performs analysis on different food products? The validation of an analysis method will ensure that our results are reliable, and due to the standardization and verification of the method, any laboratory that follows it can easily make a comparison of its results with those of another laboratory performing the same method, of course. On the scope, it includes reference methods that have been validated and also that have not been validated, as well as alternative methods validated according to part 2 of this series or similar protocols, and also methods for the analysis of samples, not only of food for human and animal consumption, but also environmental from primary production sites and production areas. It is important to mention that the number of samples for the validation and verification studies are limited, so not everything is included in the method, but we will see this in detail later in the presentation. Besides these ISO standards, other reference documents that could be important, well, of course, ISO 20468 for the validation of reference methods that we discussed previously, and especially to review food categories, Annex A, that can be found in part 2 and part 3 of these ISO 16140. It's the same annex. And as we have mentioned, in addition to food categories, so we have also feed and environmental samples included. And very important as well, so who does part 3 of ISO 16140 affect or can affect? Well, as I've mentioned before, laboratories accredited, of course, non-accredited ones as well. This would be a voluntary application that is of good practices, also relevant for certifying bodies and regulatory authorities. So I've quickly reviewed just an overview of this series of ISO 16140 standards and each of its component parts. Now we will go on to explain as best as possible what is a validation in a verification, since these are concepts that we have to have very clear in order to know in which situations one or the other applies. We have seen that a validation is different from a verification. But first of all, would we be able to say at this moment right now why it is necessary to perform the verification of a method? So to answer, I have here a very practical example with which I think the reason will be perfectly understood. Here we can see a car, so a person with a license. So we presume that they know how to drive, will get in and will be able to use it perfectly after becoming familiar with the model, etc. So there will be no problem with the car since its use has been validated. However, what would happen if a person that does not know how to drive, will the car work in the same way as with the person who does have a license? The answer is no. And that is the difference. The car works because it has been validated. However, the verification is responsible for ensuring that the user is able to use correctly the car. And it is the one that would advise us not to get into a car with a driver that doesn't know how to drive. So we have that. In other words, the validation establishes the performance characteristics of a method and objectively demonstrates that it meets the performance requirements for a specific intended use. And on the other hand, the verification demonstrates that a validated method works, implemented by a laboratory in our case, has a performance according to the specifications determined in the validation, and that it is suitable for its intended use. So now, what would be required to be able to use a method in a laboratory? So it is necessary to carry out two steps. The first is the validation, as you will surely have imagined after the previous example. Validation starts first with a study in one laboratory, in a single laboratory, followed by an interlaboratory study, which is performed by many laboratories of identical samples at the same time. And it is there where we find the scope of the method that specifies the group of products to which said method can be applied. A smaller group within this will be the scope of the validation, which will be the products for which the use of the method has been validated. And finally, an even smaller group will be the scope of the laboratory, which are the products that the laboratory analyzes routinely and are part of the scope of the validation. So it is very important to know the scope of the validation so we can choose the products to carry out the verification, as we can see here. The second step is the method verification, as we have already said, to demonstrate that the laboratory can perform the method satisfactorily according to the performance specification. But can a method that has not been validated by an interlaboratory study be verified? Some of you may already know the answer, but I will carry on. Please, if you can, leave the answer in the question box and we will get back to it later. Now, for the verification, we have also two steps that are also kind of two types of verifications. On one hand, we have the verification of the implementation to demonstrate that the laboratory is capable of carrying out the method satisfactorily. That is exactly what we have been discussing all this time. And for this, it will be necessary to use one article for each type of method, depending whether it's qualitative or quantitative, and then compare the result with the one obtained from the validation studies. The second step, second type, if you will, is the food item verification, to demonstrate that the laboratory is not only capable of performing the method, but also it declares the items to be within its scope. And for this verification, items that are routinely analyzed in the laboratory and that are, of course, within the scope of validation will be selected, as well as difficult items, which we will see later what is considered a difficult item. Again, we're going to review this issue of scopes, so there's no confusion at all. We have first the scope of the method. In it, we can find 15 different categories of products for human consumption, one category for animal nutrition, one category for environmental samples, and well, actually two categories for environmental samples, which are samples from food production and from primary production sites. Then in the scope of the validation, if it's for a broad range of foods, for example, milk and milk products, raw meat products, multi-component foods could be chosen, which are foods from different categories that are within the scope of the method. This is very important, within the scope of the method. And therefore, the scope of our laboratory, if we want to verify a broad range of foods, we should select for the first verification, the implementation verification, a product within the scope of the validation, which could be minced meat, an item that is within the raw meat category. And for the second verification, the item verification, we have to choose difficult items that are among those commonly analyzed products in the laboratory. That could be pizza for multi-component foods, black pepper, alfalfa, and so on and so on. So here you can find all the categories that we have been mentioning, 15, as you can see here, the second row, and up to here in the third row, 15 categories for of food for human consumption, one category for animal feed, and two categories of environmental samples. These categories include all kinds of food items, from the most typical ones that are commonly analyzed, such as milk, meat, eggs, fresh produce, to infant formulas, nuts, bakery products, and confectionery. We can find everything here. And so we can see more clearly to what I was referring to with category, type, and item. Categories are groups of types of food that have the same origin. Then we have a type, which are groups of items transformed in the same way with similar intrinsic characteristics and therefore a similar microbiota. And then the item, which would be the matrix. So for example, here we have a category of fresh produce and fruit that includes several types of food: cut, ready-to-eat fruits, sprouts, leafy greens, vegetables. And then in each type, we have some items. So for cut ready-to-eat fruits, we have fruit mixes. In sprouts, we have soy, alfalfa, fenugreek, mung. In leafy greens, basil, lettuce, basil, cilantro. So these are some examples that you can see. This annex is a great help when choosing the items to carry out the verification, and it also gives us information about the relevant microorganisms to analyze according to each item. So now, once the difference between the validation and verification has been reviewed, which I hope is much clearer now, we will fully enter into what interests us today, that is the verification of an alternative method in a single laboratory. So we must take into account certain general considerations. First of all, as we've seen, the validation is relevant to the verification, so any published validation reports should be consulted, especially to be able to select the appropriate item for our verification project. In the second part of the presentation, I mentioned an example of a laboratory that wanted to verify a broad range of foods, since different scenarios depending on the scope of the laboratory. So we can find verification for a broad range of foods, for a limited range of foods, and for a broad range of foods and other categories. Of course, you can have only for other categories range of foods, and you can have for a limited range of foods and other categories. So there are different situations that you can have, and in to see a few, we will get into detail shortly. And now we're going to answer this question that I asked before. I don't know if anybody answered it, but yes, it is possible to verify an unvalidated reference method, and that is Annex F4 of this part 3. You can consult it, and there you have all the information available. So we begin with the first step, the implementation verification. Remember that an item must be selected within the scope of the validation and within the scope of the laboratory. For quantitative methods, a difficult item is selected that is within the scope of the validation, even though it has not been tested during the validation. And well, here we have, just some information and a table with a summary of this and the importance of the item, depending on the type of method to be verified, if it's qualitative or quantitative. Continuing with step 2, which is the item verification, it is necessary, again, remember, to analyze a difficult item included within the scope of the validation, even if it was not analyzed during the validation, and always obviously within the scope of the laboratory. If not, this would not make any sense. So here we have the information. And now, what would be considered a difficult article? This can be found in Annex B of Part 3, and the microbial or physical chemical characteristics of this article must be taken into account, such as if any microbiota is added for a probiotic, if the sample has a high microbial load, or if its microflora may interfere with the recovery of the target microorganism. Also, its composition, pH, water activity must be taken into account. If there are any inhibitory compounds, even its physical structure, and if it has any added colorant. Also, if during the processing of the article, the manufacturing process could affect the recovery of our target microorganism. Some examples, fermented foods, cured foods, raw poultry, raw milk, and multi-component foods. So now, we will look in more detail at different laboratory scope scenarios, like I mentioned, for the verification, starting for a broad range of foods. Here we have a scheme form of the number of items required for both verifications. In this case, for the implementation verification and for the food verification. For the implementation, we select one article, and then for the item verification, at least 5 difficult items from different categories are selected, all within the scope of validation. Here we can see that all these categories are included within the scope of the validation, but not all of them are tested. Okay, so it's not obligatory that a food category is tested, but it must be included within the scope of the validation. Here we have, again, all our categories, our food categories. We have first in green, all the categories included within the scope of the method for a broad range of foods. We are excluding the other categories, animal feed and environmental samples. Then in blue, we have the categories included within the scope of the validation. In red, the ones that were tested during the validation. And finally, in orange, all the categories within the scope of the laboratory. This is a more visual way to confirm that the lab scope should always be within the validation scope. Now we have a situation where the laboratory is interested in doing a verification for a limited range of foods. And when it's limited, it should be less than 5 categories included in the scope. In this case, the categories would be limited only to those tested during the validation, as we can see here. And if the scope of the laboratory was like I've said, less than 5 categories, at least one difficult item for each category would be chosen for an item verification. How would that look like? Well, then we have here. We have this table where we can clearly see the scope of the method in green. Then we have the scope of the validation in blue. The categories with a red cycle, which are those tested during the validation, and in orange, the limited scope of the laboratory in this case. Finally, we have another situation in which we not only include a broad range of foods for human consumption, but also for animal consumption and environmental samples. And since if any of these other categories are included in the validation, and it is desired to declare them within the scope of the laboratory, an article from each category must be included for the item verification. And that is where we're really going to see a difference between these different situations, scope situations, because for the first step, for the implementation verification, the requirement will be the same in all of these. So here we have again our categories, and it changes a bit. We see the scope of the method includes these other categories. The scope of the validation includes them as well, as we can see here. Some have been tested during the validation in red, and in orange, the scope of the validation, in which we have said one article should be chosen from each of these other categories to be included for the verification. And so it can be a bit confusing with so many rules or conditions, but it really isn't. And we have a summary in this table of the minimum number of items for the verification. As we have said before, in any case, an item will be necessary for the first step, as we can see it here clearly in the first column. Then that is for the implementation verification. Then it can be a bit confusing for the item verification, but this will depend on the scope of our lab. We could sum it up as in broad range of foods, a minimum of 6 items, 1 plus 5 of at least 5 different categories, so at least 6. If the scope is limited to X number of food categories, less than 4, it would be 1 plus X number, in total maximum 5. If you include non-food categories, we would have 6 and 1 more for each of the other category in the case of being a wide range. And then if it's a limited range, we would have a maximum of 8 for the 5 plus 1 item for each other category, of which we only have 3. So there, maximum 8. And then finally, if our scope is only limited to the other categories, then a maximum of 4 items, 1 plus 3. So there you go. It looks quite confusing at first, but then it's simple, really. Another important aspect is the preparation of the target microorganisms. For the inoculations, we have, of course, artificial contamination from culture collections, which can also be internal collections, reference materials as well. It is important to select strains from matrices that belong to the same category as the article, and strains that are representative of the microbial nature that aren't the same. Using stressed cells is recommended, but not required, although it would be ideal. Then we have here the required performance characteristics to be determined for the verification. There are different ones, like we've seen, depending on the type of method, qualitative or quantitative for both verifications. So for qualitative methods, only one performance characteristic is required to be determined. We have it here, and it is the estimated LOD50 required for both the implementation verification and the food item verification. And for quantitative methods, we have 2 characteristics to be determined. The intra-laboratory reproducibility standard deviation that only applies to the implementation verification, and the estimated bias that only applies for the food item verification. So that is what we will see now, how to determine each of these performance characteristics, and we will begin with the one required for qualitative methods. We are going to go over this table again, and as we can see, we must determine the estimated LOD50 for each step of the verification. The LOD50 refers to the detection level with a 50% probability of detection. That is the detection level for which 50% of the tests would give a positive result. The LOD50 is found in the validation studies, and we compare it to the estimated LOD50, the LOD50, that we obtained during the verification. So to do so, we can find different protocols in the ISO standard, which are basically experimental designs for the laboratory to select according to their preference. So we can see here in this table 3 different protocols and inoculation levels for the test portions and the number of replicates. That is the number of test portions for the same item. To prepare depending on the protocol. For example, for the first one, we would have 1 for the high level and 4 for both the intermediate level and the low level. For protocol 3, since it's not based on different levels of inoculation, it is based on a fixed level from 3 to 5 CFU per test portion, so 7 replicates are indicated. And for all of them, as you can see here, a blank control must be chosen, that is the uninoculated level. And as I mentioned before, the LOD50 from validation studies is necessary, but this information, if it's not available, then a theoretical level of 1 CFU for test portion can be used. So let's get into the workflow of the estimated LOD50. It's quite simple, as you can see here, because it basically involves everything related to the target microorganism and the test portions, inoculating them, performing the verification method, and estimating the LOD50. So it's simple. Now, how do we determine the estimated LOD50? It is really one of the easiest parts because we will only have to look at some tables within the ISO. And in these tables, we will find the combination of all the different positive results that we can obtain. So for, for protocol 1 and 2, we must record the number of positives obtained at each level of inoculum. And then for protocol 3, the results will be evaluated based on the positives obtained from the 7 replicates. In any case, and obviously if our blind test is positive, then we would have to repeat the test again. And the limits of acceptability for each protocol, well, for the first two, the estimated LOD50 must be less than 4 times the LOD50. And for protocol 3, we must obtain at least 6 positives. In this case, we do not obtain an estimated LOD50 value as such. As an example, we have for protocol 1, we see that here we have obtained 1 positive for the high level, 4 positives for the intermediate level, and only 2 positives for the low level from the 4 replicates, and none for the blind. To obtain the estimated LOD, the table indicates that it is equal to 0.7 multiplied by the low inoculation level that we have, that is 2.2 CFU per test portion. That would give us an estimated LOD of 1.54, and in this case, it would be accepted since it is much lower than 4 times the LOD50. So the verification of this item would be complete. So let's go ahead and turn to the performance characteristics to be determined in the case of quantitative methods. We return to this table. So 2 characteristics must be determined and 1 for each type or step of the verification. So we're going to start with the first one, which is the determination of the intra-laboratory reproducibility standard deviation, or SIR. The name can throw us off at first, but really it just means that we are going to put the method to the test to challenge it. Remember that it only applies to the verification of the implementation. So again, we're going to compare what we will obtain with the interlaboratory, not interlaboratory reproducibility standard deviation that we will find in the validation studies, as this value would be SR. We are going to analyze 10 samples, and each one will be first homogenized, divided into 2 portions, tested under different conditions, meaning different personnel, batches of median reagents, instruments, equipment, and so on. And if required, the suspension can be inoculated artificially. So, as I've said, we must really test the method, verify that it works correctly, no matter how different the conditions are. In each suspension of the method will be carried out, and the quantitative results will be obtained, and then the intra-laboratory reproducibility standard deviation, the SIR, will be calculated using an equation that looks a little bit like that. And it can be done manually. There is an example of it in the ISO, but of course, there are ways to do it with Excel that are quite easy. Then we have the limit of acceptability. That is that the SIR should be or must be less than or equal to 2 times the lowest average value of the SR, which is actually the most challenging value for the method. Here we have an example of a table of SR values obtained from the validation study, we see that the lowest value of the low inoculation level is 0,18. And if our obtained SIR is equal to or less than 0,36, then we will have successfully performed the implementation verification of the method. We are going to continue with the last characteristic to determine for a quantitative method, which is the standard deviation, and it only applies to the item verification. To do this, we inoculate the article with a known level of contamination and compare it with the result of the inoculum suspension used to contaminate the initial suspension. We also have 3 levels of inoculation. Each one will be carried out in duplicate and our blank to later count both the contaminated article and the inoculum suspension, which is what we must compare. The results will be expressed in logarithm CFU per portion per test portion, and those applicable to the inocular suspension in logarithm CFU per milliliter. So this would be the workflow for the standard deviation, the e-bias, starting with a sample that is homogenized, and 7 test portions are taken, of which 1 is left uninoculated, and the remaining 6 are inoculated because it is done in duplicate. Here we have also our 3 levels of inoculation: high, intermediate, and low, which would be the ones we would find naturally in the item. And in all of them, we carried out the quantitative method to obtain the count of both the test portions and to compare them with the results of the inocula. So for this performance characteristic, the absolute difference between the results of the article and the suspension must be equal or less than 0,5 logarithm CFU, which is the acceptability limit to be met. Here we have a table with the results of the test obtained from a method to verify, and we have for the medium level, we have here all the duplicates and the results, and then in the last column, the absolute difference, that is the e-bias, and how we can see each one is less than 0,5 logarithm CFU, with which the acceptability limit would be met, and then the verification of the item would be successful. So we're going to finish the presentation quickly by closing with conclusions that we take after having reviewed part 3 of this series of ISO 16140 standards. So starting with reality, that what would take for a verification project. So for a method, of course, it would involve extra work. There's no other way to look at it. Personal resources, time resources, which are both very valuable. It would also be necessary to review the validation of the method, since we require some data. However, there are not only drawbacks, because the benefits that the verification would bring us, in my opinion, and surely many of you, exceed the cons, since the personnel necessary to carry out the verification does not require any type of expertise. It is the same personnel in charge of carrying out the analysis daily, and the verification would give us a quality assurance of the results in the short term. So what advice can we give for the verification beyond of what we've seen? Well, again, the recommendation of using stressed cultures to have a more realistic representation of the samples analyzed daily in our lab. The protocols indicate a minimum of replicas, but it would be ideal to add more to make our verification more robust. And although the only one that can carry out the verification is the laboratory, it is not possible to outsource this process. It is possible to ask for advice or help from experts in the field. So closing thoughts, the objective of the ConaLab talk was to get to know and understand fundamental concepts regarding the validation and the verification of methods, and to distinguish each one who is involved, what they contribute to our laboratory, and as well as the importance of the verification of methods. It is good to see a bit of the evolution of what was a single ISO 16140 standard published 20 years ago only for the validation of alternative methods versus the work that has been the development of this series of standards that contemplate much beyond this. And its expansion is in response to the need of laboratories to validate or verify the methods they use, since every day we find more and more products on the market. And well, thanks to this part 3, which is very necessary, will now become an indispensable tool for the standardization of method verification. And of course, in our case, it has also been a first presentation on the subject to see what would be considered interesting, whether it would be considered interesting for us to prepare a ConaLab talk as a practical case, what a verification, for example, of a conduct gene kit would look like, which is our line for the detection of pathogens by qPCR. So do not hesitate to write to us so we can prepare it and present it if it's interesting to you. And well, without any further ado, let's go into the questions that you've been sending. This ISO is for a verification in a laboratory. Could it be applied to more laboratories? So I understand that you are referring that if this ISO could be used, could be applied to multiple laboratories, and the answer is no, unfortunately no. Like I've said, the verification is not possible to outsource or contract. It would defeat the purpose of the verification because so something applied in multiple laboratories would not be a verification, since each laboratory would have to perform the verification on their own. That's why this part 3 is dedicated to a single laboratory with the products that they analyze routinely. If my laboratory is not accredited, is verification really necessary? So the short answer would be yes, although the verification would be voluntary for these laboratories. It would be recommended as a good part, as a part of good laboratory practices, because if you're not doing this type of evaluation yourself, how do you know if your results are really correct? So the verification lets you know, basically. And quickly, a last one. I added a new matrix to analyze in my lab. Do I perform the verification? If new items are added and these are from new categories, so the emphasis on new categories, then yes, the verification must be carried out for this new item. However, if the matrix belongs to one of the already verified categories, it would not be necessary. I have a couple more questions, but I'm going to end here the presentation. The time has run out. I hope this webinar has been helpful and useful to you, and that we have cleared out some doubts that you may have been having about these issues. I want you to remember that the recording of the webinar and the certificate of assistance, that you will have them tomorrow via email. The platform sends them automatically. So I invite you to follow us in our LinkedIn, and subscribe to the newsletter. So through these channels, we will let you know more about new upcoming ConaLab talks and everything that we have new. So until next time.